Looks like it's working now on YouTube, so I'm going to get started. Hello everyone, and thank you again for joining us for this week's Fine seminar. Um, my name is Lauren Hayes. I'll be the host for this week's speaker. And before I get started with the introduction, I just want to thank last week's speaker, Christine Drea, for see if I can get this, a really fascinating talk on female dominance in lemurs and meerkats. Next week, we have a really interesting panel coming up. This panel will be about writing for the public, how to communicate as a scientist with the public, obviously a very important skill set. That'll be hosted by Karsten Schroden, and the participants will be Raghavendra Gadagar from India, Joan Strassman from Washington University in St. Louis, and Matthew Cobb from the University of Manchester. Please come and attend that. It should be a fun panel discussion. This week, I'm happy to introduce our speaker, James Tumulty from Rhodes College. Rhodes College is a liberal arts college in Memphis, Tennessee, which is about, I think, 3 hours from where I am. James' history is he got his BS from Siena College, which is in New York, master's degree from East Carolina University, PhD from the University of Minnesota. He was then a visiting professor at College of William and Mary in Virginia in the United States. And he's a relatively new assistant professor, started in 2024 at Rhodes College. And so I'm really happy to be hosting him as our early career scientist or a rising star for the Fine. As you'll see from James' talk today, I imagine they're interested in animal communication, social recognition, and he uses a diversity of taxonomic groups. He's worked with frogs, but also has done work with paper wasps and salamanders. His research has been funded by the National Science Foundation and the National Institute of Health in the United States. And he's generated approximately 20 papers. And if you look at his publication record, the quality of the journals in which those papers are going are really excellent. Papers have been in behavioral ecology, current biology, animal behavior ethology, American Naturalist, PNAS, evolution, and I believe there was a tree paper in there as well. So publishing quite well at an early career stage. I was also impressed by his teaching philosophy statement, which emphasized learning conceptual frameworks, which is it's close to my heart. I try to get my students to think about things conceptually. And so it seems like he's interested in taking his work and making that into an educational experience for his students, which obviously is going to be really important at Rhodes, where I think it's mostly undergraduates. And so with that, I will stop the share and introduce the title. Today, James's title will be Evidence of a Selective Link Between Cooperation and Individual Recognition in Paper Wasps. And with that, I'll turn it over to you, James, and thanks for joining us today.
All right. Thank you very much for that. Wonderful introduction. Okay, let's see. Am I
Switch
sharing the right?
displays. Switch displays at the top, I think, is how we did it before.
Oh,
There you go. Looks great.
okay. All right. Thanks for the introduction. Thank you very much for having me. Really excited to talk with all of you. Yeah, so I'm at Rhodes College. I just got here, so I don't really have anything to talk about for my work so far here at Rhodes. I'm going to be working on communication in frogs and continuing some work with paper wasps. So what I'm going to talk about today is work I did as a postdoc in the lab of Michael Sheehan at Cornell, the Laser Lab, Laboratory for Animal Social Evolution and Recognition. You can see our excellent logo there. So these are some of the people I worked with in the Sheehan lab on this project. People in the lab and some external collaborators, Hannah Weller and Stephen Van Belleghem. So overall, I study animal social behavior and communication. I'm really interested in the ways that social environments serve as sources of selection for traits and how animals navigate their social worlds. The trait that I'm going to focus on today is social recognition. I think it's really a foundational trait for social behavior. Um, you know, just with some quick examples, you can imagine these penguins, parent penguins having to identify their offspring in a crowded environment. Recognition is key in providing care in large colonies. Scaling it up to animals that have strong dominance hierarchies, which I know you guys heard about last week. Um, Social recognition is key because it allows individuals to not only identify other individuals in the group, but also know where they are in the dominance hierarchy. Um, and sort of putting this into some general frameworks. On the one hand, we have a really influential idea in the study of social evolution, the social intelligence hypothesis. Uh, you know, generally states that social complexity is an evolutionary force driving cognitive complexity because of the cognitive traits that allow animals to navigate their social lives are assumed to be cognitively advanced and somewhat costly to maintain in terms of the brain tissue. Um, and This hypothesis yields a general expectation that as social complexity increases, cognitive complexity also increases. And a lot of tests of this hypothesis, of course, rely on proxies for these things because they're hard to measure directly. So, for example, group size as a proxy for social complexity, or brain size as a proxy for cognitive complexity. And there's a lot of good evidence for this hypothesis. There's also some evidence, you know, calling into question this hypothesis. And I think part of the issue is that, when you do rely on proxies for these traits and you're looking at correlations, there's always different ways you can parse the data. So I think, in my work and what I'll talk about today is focusing on a specific cognitive trait and trying to relate a specific cognitive trait to cooperation and to fitness. And then coming at this from a different way, we also evolutionary models for cooperation often assume individual recognition. There are, of course, models looking at cooperation between relatives that rely on kin selection, but cooperation between unrelated individuals, reciprocity requires individual recognition in order for individuals to be able to track and remember who has helped them in the past, who has not helped them in the past. Individual recognition is the key trait here. Um, so this is, putting social recognition in these frameworks. I think it's a really key trait, and also I think sort of a somewhat of a gap has been clear evidence that um, variation, excuse me, in social recognition is clearly linked to fitness in cooperative groups. And I think part of the challenge arises because you really need variation in the trait. In this case, you need variation in recognition behavior in some way, to relate that trait to fitness. Um, and so I'll also talk about social recognition as a system, and that's because it's not just the receiver. It's an interaction between a sender and a receiver. Um, and so you get a frog graphic because I also study frogs, even though I'm not going to talk about them today. So from the sender's perspective, right, individuals must be identifiable based on their phenotypes, right? So there must be variation in the signal such that the signal could be used to reliably identify the individual sender. And then from the receiver's perspective, receivers have to do a number of things. They must be able to perceive individual differences in the signal. They must be able to remember signal or cue properties, as shown here with a template in the little thought bubble of the frog. Then they have to categorize new signals they encounter based on familiarity, whether or not they match this memory or template. And finally, they respond. And that's the thing we actually measure when we measure recognition is the response. A lot of my interest in research is based around um, studying social recognition as a system and trying to understand how these different components respond to selection. Okay, so this is the system I'll talk about today. It's the northern paper wasp, Polistes fuscatus. Um, they're really interesting wasps because they are cooperative in a number of ways. I'll explain on the next slide. But what I want you to notice first is that here we have a nest. They form these open face nests where you can see all the cells. You may have seen them like around your house. They really like to make nests on the eaves of buildings. Um, and you can see a number of individuals on the nest. And the first thing I would like you to notice is that if you look at their faces, they have these color patterns, and each one is unique. This species is characterized by individual variation in color patterns, particularly color patterns on their faces. Um, and um, I mentioned they're cooperative in a number of ways. So here's a typical paper wasp life cycle. Uh, so in the spring, individuals called foundresses, or in other words, queens, will start a nest. Her daughters emerge in the summer as workers, and later in the summer into fall, the nest will produce reproductive individuals, gines and males, which then go to mate. The gines overwinter and become future foundresses themselves. So they're cooperative in the way that many social insects are cooperative in that they have workers who remain at the nest and help. But they're also cooperative in another way that makes them really interesting is that there is sometimes cooperation among foundresses. So you get multiple foundresses working together to build a nest, and they are not always related. Sometimes there are unrelated foundresses that work together at this stage. So that makes them really interesting um, for studying the evolution of cooperation. And there's been quite a lot of previous work on this species specifically by starting with Elizabeth Tibbets, who's at University of Michigan, and also work with her former student, who was my postdoc advisor, Michael Sheehan. Um, and many experiments on this species have shown that this species founds nests cooperatively, and they can recognize individuals. So these foundresses, if they form multiple foundress nests, they can individually recognize other individuals that they found the nest with. Um, experiments have shown that they do this by recognizing the color patterns on their faces, um, and that this seems to help them establish a dominance hierarchy where you have one dominant queen who does most of the reproduction, and then one or more subordinate queens. And individual recognition seems to allow these wasps to establish this dominance hierarchy and understand where the different individuals are on the hierarchy. Okay, so that's the background. Here's what I'm going to talk about today. Uh, Generally, the work I'm going to talk about is trying to test this hypothesis that there's been coevolution between individual recognition and cooperation. And I'm going to do that with three approaches. Uh, One, I'm going to look at geographic variation across the eastern US in color pattern identity signals and cooperation rates. Second, I'm going to talk about common garden behavioral assays of individual recognition and grouping behavior. And then the third part is I'm going to talk about using genomic data to look at signatures of selection on loci that we think are related to recognition. Okay, so starting with part one, the geographic lines. So I mentioned the previous work on this species. So a lot of the previous work um, has been done on northern populations. So I'm highlighting one population in uh, in New York, in Ithaca, New York. You can see some representative faces of wasps here. Their antennae have been removed so that you can see the color patterns a little bit more clearly. Um, so there's been work in New York. I have another population shown in Iowa, but a lot of Liz Tibbetts' work has been done in Michigan. And generally in northern populations, we see a lot of color pattern variation, and experiments have shown that these wasps can individually recognize each other. But as you move south, it changes a little bit. So here we're moving down through Pennsylvania into the Carolinas, and you can see a shift to the wasps becoming a lot more sort of this red-brown color. And there seems to be less variation. And then if you go to the very deep south, I have along the Gulf Coast or coastal Georgia here, I have two populations where basically everyone has the same reddish brown face. Um, so I spent a lot of time driving around collecting wasps from different locations, and this this trend seems apparent. So this is sort of the inspiration for looking at the relationship between recognition and cooperation is that there seems to be variation among populations in color pattern variation, and we know from the previous work that the color patterns are used for individual recognition. So this seems like a really good uh, system for looking at the evolutionary relationships between these traits because we have a variation among populations, seems to be variation among populations in the traits. Okay, so are they the same species? Yes, they are. So we sequenced genomes from these different populations. This is a SNP tree, single nucleotide polymorphism tree, um, and we have three species outgroups shown in blue, and all of the Fiscatus from all across the range clustered together and separate from the other species. Okay, so the first challenge here is like, okay, it seems like there's a change in variation. Take a quick sip of water. But how do we actually quantify this objectively? So this was a big challenge, something I worked a lot on. Um, and here's the method we settled on. Okay, so first we start with standardized photographs. So these have been standardized. There's slight differences in lighting, and they get standardized through the use of white standards. I'm showing the Mica toolbox as an ImageJ plugin that I use for this step. Um, then there's an alignment step, basically using landmarks. And running it through this R package patternize, we can align all of the images to each other so that they are the same size and shape and position so that we can make sure we're analyzing homologous color patterns. And I should say, the purpose of developing this pipeline is also so that we can run it through a lot of images. It needs to be somewhat automated. Um, And so what you're seeing here are regions that we don't care about have been masked out. So the eyes, for example, and the mandibles and the holes where the antenna would be have been masked out. So now we just have our regions of interest. And then from uh, a full set of images, then what we want to do is there's like a lot of variation, there's some different variation, different shades of different colors, right? And we want to be able to look at pattern specifically. So we need a way of binning sort of minor variation in the pixel values of different colors into color categories. And so what we did was from all the images, um, uh, we did a binning procedure to get the three colors that show up basically across all populations. You have like a yellow color, a reddish brown color, and a black color. And then we basically using an R package called Recolorize, we force each pixel into one of these three colors based on whichever color is nearest in color space. Okay, and so what we get are these like cartoon color patterns here, which are really just three binary layers corresponding to the three colors, right? And each pixel is a one or a zero corresponding to whether or not the color is present at that place on the face. And why this is useful is that then now we have data that we can analyze. We we can statistically quantify variation, and so to do that, we ran a principal components analysis of the entire color pattern. So incorporating all three different layers here corresponding to the three different colors, running all of that through a PCA. Uh, we got 24 statistically significant PCs. I'm just showing the first two here. And it's showing variation in what we would call the Fiscatus face space. And just to highlight a few, I'm highlighting a few color patterns here just so you can sort of see what they look like, how it spreads out the variation. And the yellow dots are highlighting a population from New York at the northern end of the range, and the red dots are highlighting a population all the way at the southern end of the range. So you can see in the New York population, there's uh, these wasps are um, spreading out more in this multi-dimensional face space than a southern population. So then all I did then was just compute the pairwise Euclidean distance between points to get a measure of face diversity. So when I do that, that's the measure of face diversity. So I did that per population, right? And so so for example, we have a southern population here in Savannah, Georgia. This is the pairwise distance between all of the faces in that population. And the trend, this measure of face diversity, distance between faces, we can see it clearly correlates with latitude. A lot of work to show what we can clearly see from looking at the images, that there is a clear increase in face diversity, face pattern diversity with latitude. Okay, and so then, so that's the signal part. Then I also took data on cooperation rates. So our measure of cooperation, so all the nest observations are shown in black dots across this map. Um, Cooperation is just measured here by number of foundresses per nest. Um, and so some of these data I gathered myself. A lot of the data just came from the literature from other studies. And here are the data showing the number of foundresses per nest, also with latitude on the x-axis here. And you can see that there's a corresponding increase with cooperation as we move north. In the deep south, the vast majority of the nests are single foundress nests. You do occasionally get multiple foundress nests in the deep south, but it's pretty rare. As you move north, you can see at the northern latitudes between 40 and 45 degrees, single foundress nests are still common, but we're approaching the point where uh, there are more, the median number is approaching two foundresses per nest. And you do find nests with quite a few foundresses in the north, so four, five, six, even seven foundresses on a single nest. Okay, so we have two measures here. Uh, Face diversity increasing with latitude, cooperation increasing with latitude, and all of this together, I think, points to cooperation driving color pattern diversity because being recognized is beneficial. That's the takeaway here, is that we think that the 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 reason diversity increases in the north is because individual recognition is important for cooperation, and being recognized by others is beneficial, favoring greater diversity. So that was part one, looking at geographic clines. I'm now going to move on to the second part, which is taking these wasps into the lab and studying their behaviors in a sort of common garden type environment. Okay, so what I'm going to focus on for this part is two representative populations. So we have this northern population from New York. You can see some representative wasps from this population, and then a southern population from Louisiana, just north of New Orleans, where there's very little color pattern variation. And you can see also, corresponding data showing that the northern wasps are more cooperative than the southern wasps in terms of number of foundresses per nest. So what I did was, there are many differences between New York and Louisiana, ecologically, environmentally, right? So what we did was we took these wasps from these two populations at the extreme ends of the decline and brought them into the lab to try to look at their behavior in the same environment. And we tried to do this in terms of as a sort of common garden experiment. So we actually collected gines in the fall after they had some time to mate in the wild and brought them and overwintered them in the lab. So they experienced the same conditions throughout the winter. And then the following spring, we tested their behavior in the lab. So the first experiment I'll talk about is an individual recognition experiment. Can these southern wasps recognize individuals is an important question we needed to answer. So this is the experiment. On day 0, our focal wasp here is brown. We have a green wasp. Two individuals encounter each other that have never encountered each other before, and they fight. Day 1 of the experiment is a familiar day, so the same two individuals fight again. Uh, on day 2 of the experiment uh, is the unfamiliar day. So now our focal wasp in brown is encountering a different individual, shown in light green, that it has never encountered before. And then we also controlled for any changes we might see through time by, on day 3, having our focal wasp again encountering the same familiar individual it had been with on day 0 and day 1. Okay, so this is the test for individual recognition while also controlling for any changes in aggression we see through time. And so here's an example video showing some of the aggressive behaviors. They're tested in a neutral arena, which is just a petri dish, and the wasps will fight with each other. In this situation, you can see the blue individual is trying to bite the other individual. So we can quantify all of these aggressive behaviors. I computed an aggression intensity index, in part because this is how it has been uh, characterized in the past, which is just the number of aggressive behaviors weighted by sort of how aggressive they are, how escalated they are, divided by the total number of behaviors. So it's sort of a rate-based metric. Um, was there a question in the chat? Should I address that or wait till the end?
We'll wait to the end on the questions in the chat.
Okay, sure. Um, And here's what the data look like. So we tested both northern and southern wasps in this experiment. So I'm showing you here broken down by each day. You can see that the northern wasps show a noticeable drop in aggression on day 2 when they re-encounter a familiar individual. Um, uh, I'm sorry. I'm just realizing that I actually explained the experiment wrong, and I'm really embarrassed about this. So when I explain the experimental design, so they encounter unfamiliar individuals on every day except for day 2 when they encounter the familiar individual. I'm sure you're all very confused right now. But day 2 is the familiar individual. They re-encounter the same individual they encountered on day 0. And then on day 3, they encounter the same familiar individual again. So if you can sort of forget everything I just told you on the experimental design slide, here is actually what happened. Okay, so what you can see for the northern wasps is that on day 2, they show a noticeable drop in aggression when they are interacting with a familiar individual, compared with their aggression to unfamiliar individuals that they encountered on day one and day three. For the southern wasps, we see no change in aggression, um, basically for any of the days, showing no evidence of individual recognition. And so what we actually, just pooling the data based on familiar versus unfamiliar individuals, um, uh, you can see pretty clearly here. Okay, so we have the northern wasps show noticeable decrease in aggression to familiar individuals compared with unfamiliar individuals, and the southern wasps show no significant difference in aggression to familiar versus unfamiliar individuals. All of this just to show that the northern wasps show evidence of individual recognition and the southern wasps do not. And it sort of makes sense. Of course, we can see that the southern ones don't have uh, any clear variation in color pattern that could be used to individually recognize nestmates. Okay, so then the next question is, if we have populations that differ in individual recognition, how does this actually impact their the social dynamics of their groups? Since we think that individual recognition is important for cooperation. So what I did was I put these wasps into groups of 4 individuals and housed them in enclosures and gave them opportunities to interact and potentially start a nest. Just a quick pan through our lab, you can see all these replicate enclosures, each with 4 wasps, given sort of the opportunity to freely interact and nest, and monitored for 2 months. So the first thing I did was look at huddling behavior. So before wasps start a nest, they may they just sort of aggregate together, what I'm calling huddling. This may be important in forming associations, nesting associations. So I just recorded at night who was huddling together. And the first thing we noticed comparing these two populations is that the southern wasps form larger huddle size on average than the northern wasps, which is sort of interesting, right? Because and seems at first to be something somewhat counterintuitive, given that I've already told you that the northern wasps were more cooperative. But what's interesting is that if you look at other measures, um, like for example, the variation in huddle size per group, we see that southern wasps show also greater variation in the size of the huddles. Um, and uh, and this sort of points to the huddling of southern wasps being just a measure almost like of how gregarious they are. They're coming together, and there's variation in how large the groups are, but they don't seem to be really stable. Um, so then, just to look at this more directly, I built social networks of these huddles. And so there are 4 individuals, 4 potential nodes in the network. The lines connecting the dots are weights. They just measure how many times 2 individuals were associating together. And just looking quickly at the networks, they do seem to be a little bit different in that the northern ones are a little bit more skewed, more individualized, and the southern ones seem to be more uh, equal or egalitarian. So to measure this, came up with a measure of how evenly relationships are distributed across the network, calling this the social network edge evenness. It's derived from a Shannon-Wiener index, and it just yeah, is a measure of how evenly the weights are distributed across the network. And indeed, we see that the southern social networks have greater edge evenness. They're sort of interacting more freely with everyone in the enclosure. And this sort of actually makes sense, I think, when you think about it a little bit more deeply, that what individual recognition does is it sort of allows wasps to discriminate between individuals, right? Whereas the southern wasps seem to be a little bit more, at this stage, a little bit more accepting of whoever, right? Individuals can come and go. Um, I just moved to the South, so I can say this is an example of Southern hospitality. Um, and so, all right, so this is the interactions before they nest. I also looked at their nesting behavior, and here's the part where, unfortunately, we have pretty small sample sizes. So I'm just going to just going to show the data and describe it, but the sample sizes are really too small for um, statistical tests. Um, so yeah, so the sample sizes here are four northern nests and seven southern nests. And they both started nests in the lab. Interestingly, they both populations formed multiple foundress nests, and the mean number of foundresses for the two populations was actually similar. So we don't see this in the common garden environment. We don't actually see the same population differences we see in nature. Um, why is this? I mean, I suspect it's because we're sort of forcing them into such a small space. There aren't really many opportunities for them to nest individually when they're in a smaller enclosure. So I suspect we're sort of forcing them to be more cooperative than they might naturally be. And I have the data here is showing the mean number of foundresses, and that the reason for that is because the number of foundresses wasn't actually the same through time. So there are individuals coming and going. You can see here's one example nest where it starts with two foundresses, then we have four, and then it sort of stabilizes on three foundresses through time. And then here's another example of a nest that's all over the place. Starts out with zero, which just means that a nest was started and then no one was sitting on it for 5 days. And then we see, every number of possible foundresses on this nest. It's very unstable. Individuals are coming and going. And then here's just a measure of that. How many times did the identity of the foundresses change through time? And here's where we do see a trend where the populations differ, is that the southern wasps uh, show more changes in who is on the nest, right? Whereas the northern uh, foundress associations are more stable. Um, and then I also looked at the how the nest developed. So, here's an example nest. You can see there are some pupa, which are the white um, caps there. Some pupa have already emerged from this nest. You can make out some larvae in there. Um, and so I looked at the size of the nest after 2 months of housing in this experiment. And the northern nests were tended to be quite a lot larger than the southern nests. Again, these are small sample sizes, so take it with a big grain of salt here. But just in terms of the number of cells per nest, we see that the northern nests were more successful. And then looking at the total biomass, just how much the nest plus any workers it produced, how much that weighed. We also see trend for the northern nests to be bigger. And notably, there were I think the interesting thing here is there were some southern nests at the bottom here who just really didn't develop at all past the egg stage. So here's one example of this nest. You can see if you can make it out, there's some eggs in that nest. Um, so they start the nest, they lay some eggs, but it never advances past that stage. And it seems what happens is the wasps are eating each other's eggs. That's a way that conflict among co-foundresses manifests itself um, in this species, is that they not only will fight with each other, they're also eat each other's eggs. So sort of usually it's dominant individuals eating the eggs of subordinates to suppress their reproduction. So all of this together points to individual recognition being a trait that really stabilizes social groups, right? We see more stable founders associations, more stable uh, networks of huddling individuals in populations that have individual recognition. And that scales up, it seems to scale up to more successful cooperative nesting. Okay, and then the last part of this is uh, looking at trying to determine if there's been selection for um, uh, individual recognition uh, by looking at loci that might be related to cognition. Um, so, individual recognition is assumed to be cognitively challenging. So if there's been um, if recognition has evolved recently or it's been especially beneficial, we would expect to see uh, signatures of selection on genes that relate to cognition. Um, and so the way this works, if you're not familiar, I didn't have a background in genomics for this project. A lot of this work was done by Sarah Miller, who's a co-author on the paper. But if you don't have a background, the way this works is it looks for signatures in the genome of selection. So here's an example. Let's say we have a population of wasps. Uh, um, Here shown in the oval up here, and then below it we have sections of DNA shown as black lines, and all the dots represent polymorphisms, single nucleotide polymorphisms in the DNA. Okay, so let's imagine that there's some trait that's beneficial, the dark green coloration. We would expect over time that dark green coloration to increase in frequency in the population. And correspondingly, the mutation, the red dot that codes for this trait, will also increase in frequency in the population. But what happens is because DNA is inherited in chromosomes in chunks, if there's selection on one loci, a lot of other nearby loci will be sort of swept along. They call this a selective sweep. Those will also increase in frequency and they'll be present in the same locations in the genome. So there are ways that you can basically scan a genome and look for evidence, look for patterns that look like this, where you have uh, all individuals in the population, or most of them, have the same polymorphisms at the same place in the genome. Uh, and so we did that for both populations, northern and southern wasps, and computed a measure of the strength of a selective sweep. It's called the composite likelihood ratio. And you can see this figure shows the two populations. The yellow is the northern wasps. This orange color is the southern wasps. And you can see that if you can make out the trace here, this shows the CLR values across the genome, higher values indicating stronger or more recent selection at that region of the genome. Um, and the numbers correspond to different linkage groups, probably chromosomes. And the first thing to take away is that there are high CLR values in both populations. There's evidence of strong selection in both populations. So how do we use this to look at the evolution of genes associated with cognition? Well, the first step was using a data set that Sarah Miller developed, which was genes that we think would be associated with visual cognition. So there are gene ontology terms. Genes have been categorized based on their phenotypic effects. So we took all the genes that had gene ontology terms associated with cognition, visual behavior, learning and memory, eye development, all traits we would think would be involved in uh, individual recognition in this species. Um, and we compared the CLR values for these genes with between populations. Uh, and there's evidence of stronger selection on these genes in northern populations than in southern populations. Um, and so the way this is visualized, I have to sort of walk you through this a little bit. So this is the residual CLR. So this is, for genes associated with visual cognition. They are ranked, right, based on their CLR values from higher to lower CLR values. And for both populations. And these are actually the residuals. So this is computed from the genes of interest that we think are associated with visual cognition. those CLR values, the difference between those and randomly selected genes in the genome. So we're sort of controlling for any other potential population differences between populations by looking at the residual CLR values. Okay. So, there are maybe limitations for looking at uh, genes using gene ontology terms. We also took a different approach. So a study by Floria Ui, she looked, she generated a data set of genes that in Polistes fuscatus are differentially expressed when these wasps are interacting with each other. She took these data that from paired wasps together, then looked in their brains what genes are upregulated or downregulated in response to social interactions. So we took that data set as a sort of independent way of looking at selection for recognition, and we see the same pattern. So again, we see that in the northern populations, there's stronger selection or more recent selection on genes that seem to be related to recognition or at least social interactions. Okay, so all of this together, again, points to stronger and more recent selection on cognition loci in northern populations. Okay, so just to summarize again, brought you through three very different types of evidence, looking at geographic variation, looking at, common garden type behavioral experiments, and then looking for signatures of selection in the genome. And all three, have their pros and cons. But I think really taking it all together, I think it provides really strong evidence that cooperation and individual recognition have co-evolved, and there's a selective link between these two traits in this species, right? Specifically, we see a relationship between cooperation and diversity, where variation is higher in populations that are more cooperative. And we also see a relationship between cooperation and social cognition, where populations that are more cooperative show evidence of individual recognition, and that enables them to sort of live in stable social groups. And there's evidence of strong or recent selection on genes associated with social cognition. And that's all I got. Be happy to take questions.
Thank you, James. Really fascinating talk. Really cool system. Um,
Should I keep sharing my screen or?
you can close the screen down, and if someone has a question
Okay.
directly to one of your figures, you can bring it back up.
Okay.
So just at this time, so now we do our discussion. So just a reminder, please, if you have a question, post a question mark in the chat, and I will call on you, and you can ask James the question you have. If you're a student or a postdoc, please put a P or S so that we can identify you as some early career. We want to put you up in the queue to ask questions. And if you're on YouTube, please just post your question in the chat, and Adriana will send it my direction and forward it to James. And I think that's it. Oh yeah, when you ask a question, please identify yourself and just tell us a little bit about where you're from and just your, one sentence what your research interests are. We're trying to build community and collaboration. So I'm going to get started. There was a question that was asked initially by Ruchira. If Ruchira is still here, you're welcome to go ahead and
I actually have quite a few questions, and can I ask more than one question?
Yeah, two or three is usually acceptable. Yeah.
Okay, sounds good. So I'm Ruchira, and I'm from India. I work on Polistes waspi. And my question here is number one, that when you did all these behavioral assays, did you use the wasps from the same nests or different nests, or did you take into consideration the nestmate recognition thing? How old were these wasps, and what are the specifications of the wasps basically used in the study? So this is my first question.
Okay. Okay. So for the behavioral experiments, what was the nest history, where nest did they come from?
Yeah,
Okay.
exactly.
So for the individual recognition experiment, every wasp that interacted in that experiment was from a different nest. So they had no previous experience with each other. Yeah.
Mm.
The when we did when we put them into housing to look at their sort of social interactions as a group, that was different. So there were the goal, that was three individuals from the same nest of origin and one from a different one. And our rationale there was that this is sort of mimics how nests often seem to establish is that co-foundresses are pretty often relatives. They're often from the same nest of origin. They start a nest together the following year. But you do find unrelated individuals occasionally co-founding nests. So that was the rationale for that, putting them together as sort of like some mostly relatives, but also a new individual. Yeah.
Okay, great. And also, when you studied the exposure to the unfamiliar wasps, I saw that you had unfamiliar wasp on one day, then familiar wasp, and then again unfamiliar wasp. Now, these two unfamiliar wasps, were they two different wasps or were they the same unfamiliar wasps? Because on the third day, the unfamiliar wasp actually becomes familiar because it had the exposure before, right? So my question is, were these two unfamiliar wasps different?
Yeah. So this is a lesson to not try to quickly make a diagram, the night before your talk, if anyone wants to take that lesson. Uh,
That-
yeah, I really messed up the explanation there. So what actually happened, yeah, so on day one, it's an unfamiliar wasp. So it's a new wasp. On day two, it's familiar wasp. That's when they re-encounter the same individual they encountered on day zero.
Zero, yeah.
Okay. And then day three, it's unfamiliar again. So it's a totally new individual. So each time it's unfamiliar, it's a new individual. It's not the same unfamiliar individual because you're exactly right. They would no longer be unfamiliar if they encountered each other again. Yeah.
Okay, just one more very quick question. Is there any species that you know where these markings are present only in one sex and not the other? Like only the males and not the females, or only the females and not the males? Is there any such polyster species that you know of?
Yeah, I mean, at least in Fiscatus, the males sort of all have the same yellow face. They have bright colors on the rest of their bodies, and there's seems to be variation on the abdomen and thorax, but the face itself, at least among males, is not variable to my eyes. Um, so yeah, I think Fiscatus would satisfy that, and that the males don't seem to have, at least on their face, identity signals. Um, because the males really aren't involved in many social interactions on the nest. Uh, yeah.
Thank you so much.
Okay, so I have a student, Michael, so I'm going to go to the student first, then come to you. So perfect. Could you please ask your question?
Hello everyone. Hope you can hear me. James, thanks for the talk. It was a good one. I really enjoyed it. I just have one question with regards to the result from the aggression intensity index. Like, have you tried to pair or to sort of like test individuals that are in between, like from the south and north? For example, you showed the graph of the latitude, the ones from the north and that's from the south, right? Surely you have ones in the middle, ones that sort of maybe have time to interact with each other. the ones from the south and the north? Like, have you tried to test them to see the aggression intensity index? How it differs from.
Uh, I'm sorry. Uh, can you clarify? Are you talking about
Can can
ones
can
that are in
can
between geographically, or?
can you please share your slides if
Sure.
possible?
Um, okay.
Can you go back?
Am I sharing the right way this time?
Yes,
Okay.
yes, can you go back?
Okay. Looking at something like
Here,
this? Or?
yeah, here. For example, have you tried to sort of like you see here we have the ones from the north of which they show like a lot of variations and the ones from the south. And when you did the test for the aggression intensity index, like you sort of measured between the ones in the south and between the ones in the north. So I'm asking like, have you tried to measure maybe one from the north side and one from the south side to see how that
Oh,
differs from? Yes.
are you talking about put them a northern wasp and a southern wasp together to fight? Is that what you're saying? Uh, I think
I think it
frozen.
froze up, unfortunately.
Okay. Um, all right. Yeah, I mean, haven't thought about that specifically. Uh, the thing that comes to mind is that so let's see, there was a recent study by Elizabeth Tibbets, who also studies these wasps, that she also has population differences, has been studying population differences in like color pattern diversity and individual recognition behavior. And her lab had done a study where they had basically showing that wasps from one population could still learn faces of wasps from a different population. So that's the only thing that comes to mind at the moment, if I understood the question right.
All right, thank you. But I didn't quite get what you said from the beginning because I kind of lost my connection. But all right, you can just
Yeah,
I can
sorry, you
stay
got
up
cut
until
off.
yeah. Yeah,
Yeah.
yes. All right, thanks. Mm.
can always come back to that if, perfect. But for now, Michael, why don't you go ahead, please?
Yeah, Michael Dabrowski, University of Bern. Thank you very much for this interesting talk. I have a question regarding your differences between the northern and southern populations, particularly regarding your experiment. Initially, in your introduction, you mentioned that for reciprocal cooperation, it is necessary that individuals recognize each other. That is only true for direct reciprocity, not for generalized reciprocity. And I wonder whether, because you found also social tendencies in your experimental southern population, greater huddling tendencies, for example, even if they were more variable, whether perhaps in the South they use a different decision rule to help anyone if helped by someone, for example, like is predicted in the generalized reciprocity framework. So the question is whether they just use a more simple decision rule because they don't have the ability to recognize each other individually due to the lack of a significant variation in their face color patterns.
Yeah, thanks. That's a good question for sure. I think there's still a lot unknown about how they actually manage these uh, cooperative associations, because they do, as we saw, they do occasionally form multiple foundress nests, and they seem to be very gregarious, as we saw in the grouping behavior. So I think a few things, just speculating here, I think it's very possible that they form some basic hierarchies based on, winner loser effects. For example, a subordinate wasp that gets beat up just maybe maintains a subordinate position, even if it can't individually identify other wasps in the nest. The other thing to say is that it's not only visual, they also have cuticular hydrocarbons. They smell each other, and that seems to be an important signal of nest identity. Like, everyone on a nest together tends to share a similar CHC profile, and that seems to be sort of like a quick, like, are you a member or not sort of cue, in addition to other things. So, just speculating, but without actually knowing how this works, I think it's totally possible that there could be a sort of CHC queue of nest identity that they use to accept nestmates, and then perhaps some simple mechanisms like winner-loser effects to maintain a two-tier hierarchy of dominant and subordinate. I don't know.
Great, thank you.
Okay, our next question is from Zulema.
Yeah, hi, I'm Sulema Tang Martinez, retired from the University of Missouri, Biology Department, University of Missouri, St. Louis, which, by the way, is where Sarah Miller is now. So it's nice to see her results, although I was pretty somewhat familiar with them just from seminars she's given in the department. So I guess I have several questions. You emphasize the importance of individual recognition by the face patterns in cooperation, but it occurs to me that it's also likely to be super important in terms of agonistic interactions, aggressive behaviors, and so on. And you use aggressive behaviors in your individual recognition test, which was really nifty. It's sort of a modification of habituation discrimination. I thought that was really cool. But my understanding, and please correct me if I'm wrong, is that when you have this multiple foundresses, that eventually one of the foundresses becomes the dominant, and then she becomes the sole reproductive after a certain point. I know that initially several of the foundresses can be laying eggs, but at some point you have some sort of hierarchy, presumably, I think, based on aggression, and then one female becomes the quote unquote queen for the entire colony. And so I would like your thoughts about that, because in my mind, individual recognition is important in social behaviors generally, and I'm including agonistic behaviors under the social category. So not just the cooperation that you see initially with the multiple foundresses. And I'm going to go ahead and ask all my questions because they're sort of related at one time, and I can remind you if you forget what I asked. The second question is, assuming that I'm correct, that eventually you get one foundress that becomes the queen for the whole colony, and the other ones become subordinates and are no longer laying eggs. How long do those individuals remember? Remember the faces of the other ones. Like if you did a test, like I can't remember how long they live, but much later, would you still find a difference in the way they treat familiars and unfamiliars based on face pattern? And then the third question, it seems to me that it might be interesting to also look at selection for the ability to recognize the patterns, to be able to distinguish between the face patterns in the northern and southern populations. Because it would seem that if in the north you have more striking differences in the faces, that wasps from those northern populations might also have evolved a greater ability to distinguish as compared to wasps from the southern population. And so I'm wondering if tests looking at whether southern population wasps can also distinguish faces of, say, northern wasps, and vice versa would be interesting. And I'm just wondering what you think about all of those things I brought up.
Okay. Let's see. Okay, so so the first question was about is this cooperation or is it really more about aggression?
No, I don't think that was actually the
Okay.
question. I think the question was more that you emphasize cooperation. And I think, I mean, I don't have any problem with your talking about cooperation and individual recognition being linked. I think that's, to me, that's very clear that it would be. My question was more about whether you could also sort of flip the coin and do the same thing and look at the importance of individual recognition in agonistic behaviors. So it seems that, I guess what I'm saying is that social behaviors in general, I would expect to be dependent on individual recognition or be linked to individual recognition. And some of those behaviors are going to be affiliative behaviors like cuddling and so on, but others are also going to be aggressive or agonistic behaviors. And I just wonder whether, to look at the broader picture, yes, cooperation obviously is extremely important and one way to look at it, but whether it might also be informative to look at the role that individual recognition plays in agonistic behavior. So it's not an either or, but rather both is what I'm suggesting.
Yeah, for sure. And I mean, all the measures are related to aggression, right? It's like the metrics of whether or not they recognize each other is that they really receive less aggression. And yeah, I mean, I guess I just would say that I think cooperation and conflict are sort of like two sides of the same coin, and a, a lot of cooperative animals are also very aggressive, right? Uh, and I guess a few things that stand out is like they are affiliate, they do engage in affiliative interactions. They, in addition to like working together to build a nest, they also, feed each other's offspring. They will, share food. I've seen wasps that like bring back a caterpillar and hand it off to another individual. So there are these sort of affiliative, helpful behaviors that they engage in with other foundresses. But the easiest way to test this is through aggressive interactions. And the overarching sort of hypothesis for how this works, again, is that it's individual recognition plays a role in the development and stability of dominance hierarchies. Being able to recognize who the dominant individual is, who are the other individuals on the nest. So in that sense, it is sort of an aggression-based social system. I don't know. I don't know if I answered your question or not, but
yeah, pretty much.
okay.
I just sort of feel, and this is in no way a criticism at all. I mean, I thought it was a fascinating lecture. I think it's a super neat system, but I just thought that sort of also acknowledging, like you talked about aggression for the individual recognition, demonstration of individual recognition, but sort of an acknowledgement, like you said, that they're, two sides of the same coin might also be helpful.
Sure.
Yeah. And so do you remember my second question, or you want me
I
to
do.
repeat it?
Let's see. Okay, I wrote it down. How long?
How
How
long do they remember? Like after things settle out and you have one reproductive left, that can, like the I don't know how long they live, for one
Yeah.
thing. The foundresses that end up losing out, do they live a long time, so that recognizing the dominant or recognizing each other would make a difference?
Um, uh, so yeah, I mean they live not quite a year.
Yeah.
Uh, if you take from there, they emerge in the fall and they start a nest the following the spring and they live through the summer. I think sometimes they die. Sometimes they die before the reproductives even emerge at the end of the colony, I think.
Right.
How long? So they're actually, Mike Sheehan, my advisor, actually did do an experiment that looked at um, uh, how long. I mean, he did an experiment that put wasps back together after they've been apart for a week, approximately,
Okay.
and they should still showed
Yeah.
evidence
Yeah.
that they still showed less aggression
Right.
to individuals they interacted with a week
Yeah.
prior.
Any longer periods than a week?
I don't think it's
Okay.
not
It
that
has to
I'm
be
aware
looked
of. I
at.
don't know that
Yeah.
anyone has ever tested longer periods than that,
Yeah.
but it's a good
Yeah.
question. Yeah.
Yeah. And then the third question was about selective pressures for being able to discriminate, find discrimination, and whether
Mhm.
there might be differences. This would be in the receivers, in northern and southern populations, and maybe testing, across populations.
Yeah, so this that I yeah that we basically the question is like if you provide receivers with the amount of identity information that would be required, you would think, for individual recognition, can they do it? And I think there's been a few different tests of the same idea, and generally the conclusion, so we haven't tested these specific populations I'm talking about. But comparing Fiscatus with different species, it seems that other species, even if if they're trained to discriminate faces
Mhm.
like Fiscatus or something like that, they're not quite as good as Fiscatus.
Okay.
And let's see, I know Liz Tibbetts did a study comparing populations, basically doing this experiment you're talking about, um, and still found that the wasps that she has in Michigan that do individual recognition, they still seem to be better than other populations. She has a population in Pennsylvania that seems to have less variation and doesn't show individual recognition.
Yeah.
Michigan wasps still seem to be better at discriminating
Yeah.
faces. So
Yeah.
it's a great idea for an experiment. Yeah.
Yeah. And I was thinking that, even though the southern populations to us, their faces look just like, humdrum brown, it would be interesting to see whether the wasps from the most discriminating northern populations might be able to see something that we don't see, which would be really fascinating.
That's true.
Yeah, but I guess an experiment like that, you might also have other confounds, other, like, pheromones that they use, or other things that might, allow them to tell differences. So I think it would be difficult to design an experiment that would clearly distinguish between if they're recognizing by the face patterns or using something else. But I think it'd be lots of fun to try.
Yeah, well, I'll just add that the experiment I did is one way to test for it, like interactions between actual living wasps. But a
Yes.
lot of what Liz Tibbetts' lab has been doing a lot of these sort of experiments where rather than looking at interactions between wasps, they actually do a training thing where the wasps have to discriminate between images of faces, um, and that's a way of getting at
Yeah,
more directly at
absolutely.
is it
Yeah.
just the color pattern as
Yeah.
opposed to other cues? Yeah.
Yeah. Well, thank you. It's a really, really nice talk.
Thanks.
Okay, so next we'll go to Joan. Joan, you there? There you are.
Yeah. Yeah, that was a fun talk. My question is just one for a little bit. Oh, I'm at Washington University in St. Louis. I work on social evolution, wasps, including Polistes, for 25 years, then social microbes, and now birds and social microbes. In Michigan, if I'm not mistaken, until the invasive European wasps came in, Polistes fuscatus was really the only wasp there. I don't know about New York, but maybe the same. In Louisiana, by contrast, it's my experience that Polistes fuscatus is rare and that there's at least half a dozen other species of Polistes that are much more common. So if a wasp is rarer, it's much less likely to be multiply founded because they tend to go back to the same places and stuff. So I wondered if you could tell us a little bit about the habitats where you found the different wasps and comment on their rarity and whether you think that matters.
Yeah, that I think your description is accurate. Yeah, so at the farthest north climates in North America, it's pretty much just Fuscatus. In New York, we do have a few other species there. We do have Dominula, like you said. Occasionally, you get a species called Dorsalis. But yeah, in the deep south, there are many more species. Um, uh, so I think with respect to um, are you sort of thinking that maybe some competition over nesting sites could have something to do with cooperation, or uh, I'm just trying to follow your train of thought here.
I'm thinking that in the north, Polistes fuscatus will re-nest in areas where it's nested before, becomes common. You get multiple foundresses of sisters. I just don't know what it's like in the south where they're really rare. So I wondered if you want to tell us about
I
that.
see. Okay. Yeah. You know, they do seem to be less common in the South, but collecting them, I have found a few spots where they're locally pretty abundant. So some of the spots I collected, it's sort of like this is a very quick impression, and it may not be accurate, but I felt like there was almost a gap where you get sort of down to the Southern Appalachian Mountains and Fuscatus are common, and then there's sort of almost like a gap through the coastal region. But then when you get right down to the coast, I found that Fuscatus were more common, especially along the Gulf Coast near New Orleans
So are you collecting them from house eaves or putting out nest boxes, or where are you finding
Oh
them?
yeah, they're just on mostly from parks, on the eaves of buildings at parks is the most common collection location. Uh, Gazebos and whatnot. Yeah. Um, and certainly there are other species around. There does seem to be species differences though in nesting preferences. Some of the really common species in the South, like exclamens, seem like they nest in different sorts of habitat than Fuscatus. Um, so I, you know, honestly don't have a good sense of how much competition there is between species for nesting habitats. I'm sure there's some, but I don't have something definitive to say about it. Yeah.
So the nests of fuscatus in gazebos are going to be there year after year, nesting near the parental nests, and yet they still don't form very many multiple female nests?
Across years, I don't know. Anecdotally, I collected, the 2 years that I went to collect in these southern spots, found them in generally the same locations. I never looked at like if are they in the exact same spot as they were the previous year. Um, uh, so, but yeah, across years they seem to be in the sites where I collected them. They seem to be common in the same general areas across years.
Okay, thanks.
Sure.
Okay, our next question, before I go to Adriana, if you're on YouTube, please, feel free to post a question. Adriana, forward that to me. And there's still time if people want to have follow-up questions. So Adriana, please go ahead.
Thank you, Lauren. So far, there's no questions on YouTube. Hi, everyone. My name is Adriana Maldonado Chaparro. I'm professor at Universidad del Rosario, and I'm mostly interested in social behavior and social structure specifically. I have one methodological question, and the other one, it's more on the concepts or idea of social recognition. So the first one, very straightforward. How long did you let the individuals interact within the recognition test? So were they exposed to each other 1 minute, 5 minutes?
Oh, yeah. No, it was we scored data for the first 15 minutes of the trial. So, and I'm trying to remember if it was, 30 or 45 minutes. I'm not sure of the exact duration. But the aggression data we recorded was from the first 15 minutes of the trial. Yeah.
Okay. And within the test, did you only recorded aggressive interactions? So today, I guess my question goes to the fact that individuals also express investigation. I don't know if you recorded that.
Yes. So we did. So we recorded what I called huddling, which is just when they're, in proximity but not interacting aggressively. Because they do that as well in these sort of petri dish individual recognition experiments. They also antenate each other. They probe each other with their antenna, which so the huddling behavior itself was not an aggressive behavior. So that went into the denominator of the aggression index. So it was a time when they were interacting but not aggressive. So yeah.
Yeah.
And then the aggressive behaviors were sort of weighted according to how seemingly aggressive they were, which is mostly just following previous studies on this species and other wasps. Something like wrestling is weighted a 4 because it's assumed to be more aggressive than like antenation, or they sometimes just sort of like dart at each other. That would be like I think that was a 1 on the aggression index. Yeah.
Cool. And my second question, it's on the side of, you mentioned that there were other cues for social recognition. So there was a chemical profile that sort of identified the nest. How can you tell apart in an experimental setup the recognition, the visual cue from the chemical cue?
Yeah. I mean, in this experiment, in the individual recognition experiment I ran, you can't. It's just freely interacting wasps.
Okay.
So they are certainly smelling each other in that environment. So there are many cues they may be paying attention to. The reason we know that this species can recognize color patterns in faces is because of previous experiments that have also looked at just the color pattern cue. So work by Liz Tibbetts and Mike Sheehan, where they basically trained wasps to discriminate between faces. And all the wasps see is a picture of a face of another wasp, and they have to learn to go to one side of an arena or the other to avoid getting shocked. So because of those experiments, we know that at least in these northern populations that they can individually recognize just based on the color patterns.
On the color pattern. Okay.
Cool.
That's pretty cool.
Yeah.
And just a follow-up on that. So if they have this chemical profile that identify a nest, when individuals get recruited, do they, I mean, they bring a different chemical profile, right? So is there like a convergence on the chemical cue that identify them, or how does it happen? Do you know?
That's a great question. I'm really not the person to answer this, and I'm hesitant that I'm sort of getting out of my wheelhouse here. But it's my understanding that there is some level of convergence in because they're all sort of interacting with each other and on the nests, and these CHCs can sort of get they're very they can get like rubbed off somehow. I don't understand how this works. I'm way out of my wheelhouse, but is my understanding that there's some level of convergence, even though, of course, you would expect that relatives would smell more similar to each other than non-relatives. But yeah.
Yeah, that's pretty cool. Do they use that chemical to sort of mark territories or anything like that, or is just like a personal signature?
It seems sort of like a personal / nest signature. They don't seem to be very territorial outside of the nest itself. You'll see two nests right next to each other. And the wasps are just coexisting on their different nests, but very close proximity to another nest. And they don't really respond aggressively until someone comes onto the nest itself.
Okay.
Yeah.
Oh, pretty cool. Thank you. Great talk.
All right, so I'm gonna jump to David because he's got a new question. Then I'll come back to you, Ruchira. Okay. So David, please go ahead.
Hi, Dave Quiller, also Washington University in St. Louis. If your hypothesis that the individual recognition is centered on sort of founders number issues is true, I'm curious about what's going on with workers. So do you see the same variation in facial patterns? Do you see the same geographical trend? And if so, what's going on with the workers? Is it just a carryover from the queen or what?
My guess is it's mostly a carryover from the queen, because there have been some experiments that have tested individual, let's see, trying to think off the top of my head. There have been some tests that show that workers can learn faces. Maybe they don't do it as well, and then that if you change the color pattern of a worker, it will receive more aggression. Whether that's a carryover from the queen or not is hard to say. That would be my guess, is that it's the key stage at which individual recognition is important is at the founder stage, when you have foundresses working on a dominance hierarchy, and then at the worker stage, there's so many individuals that it seems like sort of CHC signals of nest identity are probably the main sort of cue to recognize accepting a worker as being part of the nest or not. But I don't know. It's hard to say, I guess, how important individual recognition is at the worker stage to the social dynamics of the group. And I don't have a clear answer. Yeah.
The trait does not carry over to males, you said, but then males are more different, so that's perhaps expectable.
Yeah, males, yeah, like their faces all are sort of yellow, and I don't know. It's been hypothesized this may play a role in lecking behavior, whether trying to attract females. It doesn't seem like they're doing much in terms of sort of social interactions on the nest. Yeah.
Thanks.
Okay, so Ruchira, please.
Yeah, hi. So I have another question. For your northern and southern population, did you check the CO1 gene sequencing? I mean, the reason I'm asking this is, as I told you, that I work on Polistes watii. And when I first started working on this wasp, everyone told me around that this is actually Polistes olivaceus. And the difference between Polistes olivaceus and Polistes watii are in the markings. They look very similar to each other. Their colorations are same, but olivaceous has a lot of black markings on them, while watii is completely blank. The females are completely blank, at least. So that's how we, and when we did the CO1 gene sequencing, we found that they are very close, but they are two different species. And there were some morphological reasonings too. So my question is that did you ever see the northern and southern population and compare the CO1 gene sequencing?
Yeah, so that was a important like first step is ensuring that they are the same species. And so yeah, we did sequencing and made a SNP phylogeny and because the southern wasps are actually hard especially to recognize because there's to identify is there's a Polistes metricus is another species that is in the same area and they look very similar. It's there have been a number of times where I've collected wasps and I've not been able to identify it to the species level, and then we confirm with sequencing afterwards. So it is they are very similar in appearance, but we did sequence and make a phylogeny, and sure enough, they do. The Polistes from these Fiscatus from these southern populations do cluster out with all the other Fiscatus samples we've collected across the range and cluster out separate from these other closely related species like Polistes metricus. Yeah.
Thank you.
I don't see any more questions in the chat. If anyone have a follow-up they want to ask? If not, I think I'll stop it there. James, thank you. That was fantastic. Thanks for great talk and a great discussion as always. Next week, for those of you coming next week, we hope you come. There will be a panel discussion on writing to the general public that will be hosted by Carson Trodden. With that, I'm going to